Cellular respiration is a metabolic pathway whose end result is releasing energy that is used by cells to perform their task. Cellular respiration involves organic molecules like glucose. There are 4 stages of the cellular respiration: glycolysis, acetyl-CoA synthesis, citric acid cycle, and electron transport chain. Oh no, I'm hungry again. I guess I have to eat. Glycogen provides a source of glucose 6-phosphate to feed glycolysis when my levels of blood sugar is low. Glucose is a fuel molecule that is readily available in many organism cells. Glucose is not only the sugar that can be used in this process, however, it is particularly important because the brain and red blood cells only use glucose as a source of energy. In glycolysis, there are 10 reactions total and 3 main phases. The first phase is known as preparatory phase, starting with the consumption of 2 ATP molecules. I personally like to refer to this stage as the investment phase because from the get-go, 2 ATP molecules are required to help move the process along. In steps 1 and 3 of phase 1, a phosphate is broken, releasing energy in their bonds. The phosphorylation related glucose from exiting the cell, and due to the negatively charged phosphate groups in proximity, destabilizes the molecule so that it can be broken apart. Reactions 4 and 5 in glycolysis represent the second phase, the cleavage phase. The cleavage phase is the splitting of 2 sugars. In reaction 3, fructose 6-phosphate is converted into fructose 1,6-biphosphate. This step is requiring energy and is catalyzed by an enzyme known as fructokinase or PFK1. PFK1 is an enzyme that regulates the rate of glycolysis. In reactions 4, the 6-carbon molecule fructose 1,6-biphosphate is split into 3 carbon molecules, glyceraldehyde 3-phosphate and the isomer dihydroxide acetone phosphate. In reaction 5, dihydroxide phosphate is converted to 2-glyceraldehyde 3-phosphate. The final stage in this step, 6 through 10, it's sometimes known as the payoff stage because in steps 7 and 10 results in 2 ATP produced. In addition, NADH is produced. Finally, the end result of this stage is the production of 2 molecules of pyruvate. This is the image of the mitochondria. It is relevant because this is the location of the concluding steps in the biochemical process. One can describe the mitochondria as a rod-shaped organelle. The inner space is known as the matrix. The protruding parts that share the matrix is known as crystallon. The outer matrix and the inner matrix can be made in comparison to the cell wall and plasma membrane of a plant cell, just speaking in terms of position and not function. Lastly, I would like to point out that the space between the inner and outer membrane is known as intermembrane space. First, pyruvate enters the matrix through a transport protein. Immediately following the entrance, part of the pyruvate molecule is oxidized and splits off to form carbon dioxide. The second step is the NAD plus a hydrogen protein is reduced to NADH. The remaining part, CH3OC, still has a large amount of energy that can be harnessed. It is transferred to coenzyme A, a molecule that carries the acetyl group to the next set of reactions. All of these reactions are catalyzed by an enzyme called pyruvate dehydrogenase complex. Pyruvate dehydrogenase is a large complex made up of many copies of 3 enzymes, E1, E2, and E3. Pyruvate dehydrogenase produces NADH. Pyruvate dehydrogenase catalyzes oxidative decarboxylation. This process results in the formation of 2 molecules of carbon dioxide, 2 molecules of NADH, and 2 molecules of acetyl-CoA. Acetyl-CoA is a 2-carbon substrate of the first step in the citric acid cycle. Acetyl-CoA is down 1 carbon due to the decarboxylation resulting in a CO2. The third step of cellular respiration is known as the citric acid cycle, and it takes place in the mitochondrial matrix. In this step of cellular respiration, the fuel molecules are completely oxidized. There are 8 reactions that take place, starting with oxaloacetate. These reactions occur twice, as there are 2 acetyl-CoA. Secondly, a 6-carbon molecule of citrate is synthesized to produce a 6-carbon molecule known as cis-aconitate. Cis-aconitate then becomes another 6-carbon molecule known as isotrate. We lose a carbon as carbon dioxide and NADH are produced. Now we are working with a 5-carbon molecule known as alpha-ketoglycerate. Once more, we lose a carbon to carbon dioxide and gain another NADH, hence we are left with a 4-carbon molecule known as succinyl-CoA. Succinyl-CoA is now converted to succinate, another 4-carbon molecule, and GTP is produced, which is equal to ATP. Reaction 5 is a substrate level phosphorylation. Reaction 5 is the only substrate level phosphorylation in the citric acid cycle. Moving on to step 6, where succinate is converted to fumarate while an FADH2 is produced. Moving on to step 6, where succinate is converted to fumarate while an FADH2 is produced. From there, malate is produced. Malate completes the cycle and becomes oxaloacetate, meanwhile producing the final NADH in the process. The last reaction of the cycle generates a molecule of acetate, allowing the cycle to continue. So in the citric acid cycle, it results in 2 molecules of ATP, 6 molecules of NADH, and 2 molecules of FADH2. There are many enzymes involved that help synthesize each step of the citric acid cycle, beginning with the first enzyme known as citrate synthase that produces citrate from oxaloacetate and acetyl-CoA. The second enzyme is known as aconitase that produces cis-aconitase from citrate. The third enzyme is again aconitase, which produces isocitrate. Then next we have isocitrate dehydrogenase that produces NADH and releases the carbon by producing a CO2. The next enzyme we have is alpha-ketoglycerate dehydrogenase complex, which produces the second NADH in the conversion to succinyl-CoA. Succinyl-CoA synthesis catalyzes the conversion from succinyl-CoA to succinate. Then comes another dehydrogenase enzyme, succinate dehydrogenase, which produces FADH2. As succinate is converted to fumarate, fumarase converts fumarate to malate. The final enzyme in the cycle is another dehydrogenase, malate dehydrogenase, that produces NADH while producing oxaloacetate from malate. The citric acid cycle results in a complete oxidation of the acetyl group from acetyl-CoA. The citric acid cycle produces a large quantity of electron carriers, NADH and FADH, that are critical for the final stage of the cellular respiration, the electron transport chain. The final stage in the cellular respiration is the electron transport chain. It is composed of proteins embedded in the membrane and capable of transferring high-energy electrons from one protein to the next. There are 4 protein complexes that are embedded in the mitochondrial membrane. Electrons will enter the transport chain through either protein complex 1 or 2. More specifically, NADH gives up its high energy electron in protein complex 1, and FADH2 will give up its high energy electron in protein complex 2. The electron from NADH are transferred to the electron carrier coenzyme Q. Meanwhile, the hydrogen protons go into the intermembrane space. Coenzyme Q then carries the electrons to complex 2. As the electron moves from the second complex to the cytochrome C, more protons are carried from the inside to the outside of the membrane. Meanwhile, the same thing is occurring with FADH2, except starting in complex 2 instead of 1. Cytochrome C carries electrons to the 4th complex, oxidase complex, in which more protons are still pumping in the intramembrane space. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen. Oxygen is the final electron acceptor and water is the product. The protons that are transferred to the intramembrane space create a gradient that has potential energy to synthesize ATP, a special transmembrane channel protein called ATP synthase. The movement down the enzyme must be coupled with the synthesis of ATP. ATP synthase is another enzyme that are composed of two subunits, FO and F1. FO is simply the channel in which protons flow in through. F1 is simply the catalytic unit that synthesizes ATP. For every 4 protons that go through the ATP synthase, 1 ATP is produced. Plants' growth is dependent on inputs of energy, water, and CO2. Photosynthesis is the chemical process by which plants are able to use these key resources in order to thrive. Glucose is a molecule that is a building block of plants, but plants have a remarkable ability of harnessing energy from the sun and converting it to organic compounds like carbohydrates. Sunlight has both a wave and particle nature. These particles are known as photons. Photons move along a path called wavelengths. Photosynthesis is a redox reaction. The oxidation of water with O2 produced as a byproduct and the reduction of CO2 in the form of carbohydrates. In photosynthesis, the ultimate electron donor is water. Photosynthesis consists of 2 sets of reactions, the light harvesting reaction and the Calvin cycle. Sunlight is where the process begins using a proton pigment complex known as photosystems. Photosystems use absorbed light to drive redox reactions and thereby set photosynthetic electron transport chain in motion. In turn, the movement of electrons through this chain is used to drive the synthesis of ATP and NADH. Lastly, ATP and NADPH are energy sources needed to synthesize carbohydrates using CO2 in the process called Calvin cycle. Photosynthesis occurs in the chloroplast. The chloroplasts have both inner and outer membrane. In the center of the chloroplast is a third highly folded membrane known as the thylakoid membrane. The thylakoid membrane is where the photosynthetic electron transport chain occurs. The thylakoid membrane encloses a fluid-filled interior compartment called lumen. The region surrounding the thylakoid membrane is called the stroma. Carbohydrate synthesis occurs in the stroma, the light-dependent reaction in which photons and water are used, making oxygen the byproduct. In this step, it produces ATP and NADPH. The NADPH a reducing agent because it is able to give away hydrogen and the electron associated with the hydrogen. Light energy is converted to chemical energy in the light reactions. Photosystems are a large complex of proteins and chlorophyll that capture light energy. There are 4 main complexes in ATP synthase. The main complexes in the photosynthetic transport chain are photosystems 2, cytochrome complex, photosystem 1, and lastly, ferredoxin NADP reductase. An electron transport chain connects the 2 systems. In this chain is a small moving electron carrier that transports electrons from one large complex to the next. The step 1 in the process deals with photosystems 2. It absorbs light energy, meanwhile exciting electrons that enter the chain. When it loses an electron by oxidation, it is able to pull electrons from water, creating oxygen as a byproduct. The energized electrons continues to flow down the chain, releasing energy that is used to pump hydrogen ions into the thylakoid. In photosystem 1, again light energy excites electrons, but this time the electrons are captured by an electron carrier molecule, NADPH. The high concentration of the hydrogen ions inside the thylakoid powers ATP synthase. Again, ATP synthase has 2 subunits, FO and F1, with the same function as in the previous electron transport chain. The light reaction in the thylakoid have now produced 2 energy products, ATP and NADPH, which are essential in the production of sugar in the Calvin cycle. The Calvin cycle is the process following the photosynthetic electron transport chain and is the second process in photosynthesis. The Calvin cycle is when carbon dioxide is reduced to synthesize carbohydrates and is powered by ATP and NADPH. This process takes place outside the thylakoid in the stroma. The Calvin cycle consists of 15 chemical reactions that synthesize carbohydrates from CO2. These 15 reactions can be divided into 3 main steps, the first being carboxylation, the second reduction, and third regeneration. Before rubisco can act as a carboxylase, RUBP and CO2 must occupy the active site. Once the active site is occupied, carboxylation proceeds spontaneously. In carboxylation reaction, one carbon atom from a CO2 molecule is attached to a 5-carbon RUBP chain to build a 6-carbon molecule. This process is initiated by an enzyme known as ribisco. The products of the reaction result in 2 3-carbon molecules, 3-phosphoglycerate, or 3-PGA. The enzyme ribisco will be seen throughout the Calvin cycle. Ribisco is surprisingly slow. Due to this fact, photosynthetic cells must make up for this by producing large amounts of this enzyme. Ribisco is the most abundant protein on Earth and constitutes as much as half of the total proteins found in the leaf. For this energy increase to take place, the carbon compounds found in ribisco must be reduced. The second step is known as reduction. Reduction is a sequence of reactions in which NADPH acts as a reducing agent. NADPH transfers the energy and electrons that allow carbohydrates to synthesize from CO2. NADPH is just produced from the photosynthetic electron transport chain. The reduction of 3-PGA involves 2 steps. Step 1 is ATP is used to phosphorylate 3-PGA, and step 2, NADPH transfers 2 high-energy electrons to the phosphorylated compound. Creation of 1 molecule of CO2, 2 ATP and 2 NADH are required for each molecule of CO2 incorporated by ribisco. NADPH provides most of the energy that are in the bonds of carbohydrate molecules. GAP and DHAP constitutes a pool of 3-carbon carbohydrates molecules known as the triose phosphate. Triose phosphates are the true products of the Calvin cycle because they are the molecules exported from the chloroplast. For every 6 triose phosphate molecules that are produced, only 1 can be withdrawn from the Calvin cycle. This will make it possible for the Calvin cycle to continue. Finally, the last step of the Calvin cycle is regeneration. Regeneration involves RUBP and requires ATP. 12 out of the 15 steps of the Calvin cycle make up this step alone. In this step, there is a lot of reshuffling of carbon atoms that are necessary to produce 2 5-carbon RUBP molecules from 5 3-carbon triose phosphate molecules. The Calvin cycle's total energy requirements are 2 molecules of NADPH and 3 molecules of ATP for each molecule of CO2 incorporated by ribisco. NADPH and ATP play distinct roles in the formation of carbohydrates from CO2. NADPH provides the energy that is incorporated into the higher energy bonds that catalyze carbohydrate molecules, whereas ATP provides the energy needed to regenerate RUBP. The Calvin cycle does not utilize sunlight and is nicknamed the dark cycle due to that fact, but it still requires energy. Cyclic electron transport increases the production of ATP. Light is shared unevenly between the 2 photosystems. However, as light levels increase, the light energy absorbed begins to overwhelm the capacity of the Calvin cycle to make use of NADPH. This can be dangerous, but luckily there's a preventative. Electrons will then become shuffled into an alternative pathway that increases the production of ATP while slowing the production of NADPH. Electrons from photosystem 1 are redirected and from ferredoxin back into the electron transport chain. These electrons re-enter the photosynthetic electron transport chain upstream of the cytochrome B6F proton pump. Because these electrons eventually return to photosystem 1, this alternative pathway is referred to as a cyclic electron transport. Cyclic electron transport leads to an increased production of ATP. In addition, a subset of photosystem 2 migrates through the thylakoid membrane to become associated with photosystem 1. In photorespiration, there is no ATP production. Instead, ATP is consumed in 2-phosphoglycerate into 3-PGA, which can re-enter the Calvin cycle. Carbon and energy are lost when ribisco acts as an oxygenase in this process.